So the next question, 16.12, describe the role of attenuation in the regulation of tryptophan biosynthesis. So I like the attenuator system in prokaryotes because it's a nice way of showing how cells possess different mechanisms to regulate gene expression, and some of those mechanisms are built right into the RNA. So let's take a look at the trip operon. Just briefly here, this is TRPR, the TRPR repressor, which makes more sense than LAC-I, which is why, if you were reviewing some of the earlier problems, I was calling the LAC-I repressor LAC-R, but no. LAC-I is LAC-I, TRPR is TRPR. They're both the repressors of their respective operons. And they work in similar ways. So similar, in fact, that we can use the same conceptual diagrams of the proteins. So right now I'm diagramming the promoter, the operator, and we have something extra in the trip operon. We have the leader sequence, L, with the attenuator region right here, and that's going to lead into trip E, D, C. This is easy. Backwards alphabetical order. I think there's a special name for that. I forget what it is though. Okay, these are the structural genes. Structural genes here. And okay, structural genes are any gene that is not involved with regulating gene expression. And sometimes we call the trip promoter region trip P and the trip operator trip O, and sometimes we'll call the leader trip L. If you call it trip L, that's okay. It's okay with me. Okay, so L is also known as the leader region, or sequence is also fine. Leader region, leader sequence, they are both good. Okay, now the easy thing to know about the TRP repressor is, well, it is a repressible system, unlike the LAC operon, which is an inducible system. This is a repressible system, and what does that mean? So that means unless tryptophan is around, tryptophan is an amino acid. So here's our tryptophan. So tryptophan, no E at the end there. When tryptophan is present in the cell, it is the effector molecule. It will bind to the TRPR repressor. So here's the repressor with no tryptophan bound. Here's the tryptophan binding site down at the bottom here. And you'll notice when there's no tryptophan bound to there, we can't bind the operator. So no tryptophan bound. RNA polymerase can come in and transcribe all of these biosynthetic genes. And biosynthetic genes, these are genes which encode biosynthetic enzymes, which are synthesizing a biochemical like tryptophan. So they're all part of the tryptophan pathway. Well, we can assume they're all part of the tryptophan pathway, going from compound 1 to 2 to 3 to 4 to tryptophan. Okay, and this might be E, and this might be D, C, B, and so on and so forth. So no tryptophan in the cell. It will not be bound to the repressor, and as such, the repressor will not be bound to the operator. The operon will be transcribed. Okay, tryptophan binds here. Let's say there's plenty of tryptophan in the cell. Tryptophan binds there, allosteric shift causes the operator binding site to appear. Now it can bind to the operator and it can block transcription. So this right here, when the operator is bound, when the repressor is bound to the operator here, we get a 70-fold reduction in transcription of the structural genes. Okay, so tryptophan is present in the cell, binds to the repressor can now bind to the operator, and we get a 70-fold reduction in transcription of these. So there's still a little bit of transcription. Sometimes the RNA polymerase makes it by the TRPR repressor, but for the most part we get a major reduction in the amount of transcription of the operon. Okay, so now there's still some transcript being made, and we can decrease transcription even more using the attenuator system. So how does that work? Okay, we are going to focus on this part right here. Okay, so we have a very small amount of transcription occurring on of the trip operon now. So RNA polymerase is coming in here, and it's starting to transcribe. And okay, we're transcribing at the leader sequence. Okay, let's take a look at the leader now. So what is in the leader sequence? Well, let me go back to the yellow pen here. Okay, so RNA polymerase is transcribing. We're going to have a sequence here called sequence 1, sequence here called sequence 2, and if you remember from class, we are going to have 4 of these little sequences here. We don't need to know what the sequences are, except for right here, we're going to have a 5th sequence that is uracil rich. So a U-rich sequence right here, and this is called the leader sequence. Sequence here, leader sequence. That's spelled wrong, isn't it? That looks better. Okay, so RNA polymerase is, transcribing here, and again, it's happy. It likes to transcribe, and looks like a fish a little bit, doesn't it? With bones, a fish head with some fish bones. Anyways, this is not a fish head with fish bones. It is an RNA polymerase, and this is the leader sequence. Okay. Now, one thing we need to remember is translation can happen at the same time as transcription in prokaryotes, because translation is there's no nucleus, right? So everything's in the same place. DNA is being transcribed in the same place that RNA is being translated. So a ribosome could jump on here onto a Shine-Delgarno sequence here and start translating. But we're going to take a look at the situation where there's no translation going on. This is just being transcribed. Okay, so no ribosome latches on to this leader sequence. What's going to happen? Well, it turns out that these are complementary to one another. The sequences are complementary. So are these right here. And 2 and 3 are also complementary, but we don't need to worry about those just yet. So when there's no translation going on, the RNA is going to form this secondary structure with 2 hairpins. So we have the no-name loop here. You could call it 1 and 2 if you want. Loop 1 and 2. But this loop right here has a name. It is called the Terminator Loop, or Terminator hairpin, or Terminator stem loop, whatever you want. It's got Terminator in the name. And it is made of sequences 3 and 4. 3 and 4 are complementary to one another. and they will form this loop right here. 1 and 2 form a loop, 3 and 4 form a loop. They form the Terminator loop, and when they form that loop, they are going to knock this RNA polymerase right off of the DNA template. Right? So, I like giving my RNA polymerases personalities. Okay, so they knocked it right off. What knocked it off? This did right here. Somehow formation of this loop causes the RNA polymerase to be knocked off the transcript. Okay, so as a result, what happens? So we don't transcribe EDCBA genes, which are needed to make tryptophan. So no tryptophan is being made. Okay, so there's another situation. Let me erase this stuff here and start over. So another situation. That situation right there was when no ribosomes were present for some reason, or no ribosomes latched on to that transcript and started translating. So we have two other situations where ribosomes attach and start translating. Okay, let's look at the first one. So the first one is going to be, okay, so we have, remember 1 and 2, in the previous example, there was no ribosome present, so nothing was being translated. In this one, we have a ribosome, and it has bound, and it is starting to translate this messenger RNA. And what it's going to translate is called the leader peptide. So we have a leader sequence, and now we have a leader peptide. And it's just going to be a short peptide, couple, I'm not sure how many amino acids, but short, just made from codons contained in the leader region. And here's our A site, P site, E site, and it is translating. We have a little dipeptide, no, not dipeptide, but a little peptide being made. And you can see sequences 1 and 2 are occupied by the ribosome. And because they're occupied by the ribosome, they can't form that little hairpin loop. 3 and 4, however, can form loop, the Terminator loop still. And then we have the U-rich sequence. And because this Terminator loop can form, well, the RNA polymerase is going to get knocked off the DNA template. Again. I'm sure he yells much louder than that. But, okay. He or she falls off the DNA template. And I could draw the DNA template here, couldn't I? Okay, so an important aspect of this is that the ribosome does not stall. So no stalling, no pausing. It has no trouble making this peptide here, this leader peptide. No trouble. So no pausing. It has everything it needs to make that leader peptide. And if it doesn't pause at all, well then we can't get transcription of the biosynthetic genes. It has to pause. Okay, and we're going to look at the case where it does pause down here. We're going to learn why it does pause in this third example. Okay, so here's sequence 1 right here. And now what we have is our ribosome. It's bound, it's translating. Okay. Bound and translating the messenger RNA. My A, P, and E sites got pretty small here. Okay, it's translating, but something happens. Okay, so in here we have a couple of codons for tryptophan. Okay? Now, if tryptophan levels are low in the cell, what else will be low? The levels of charged tryptophan tRNAs, right? So tryptophan. So, and this would be, what would this be? This would be CCA. Okay, so if these levels, these are charged tRNA trip. Okay, if these levels are low, there's no charged, not enough charged tRNAs in the cell. What's going to happen is when this ribosome gets to these trip codons, well, it's going to pause. It's going to have to pause, and it's going to have to wait for the elongation factor. If you're remembering, elongation factor TU transports the charged tRNAs to the codons in the A site. You know, if it has to wait, well, then the ribosome will pause or stall right there, and as a result, this little 2 sequence here, which is also complementary to sequence 3, well, this will form right here. And this loop right here is called the anti-terminator loop. Anti-terminator loop. Okay, so 2 and 3 forms the anti-terminator loop. 4 does nothing. It's sad. But RNA polymerase now is happy because it can keep transcribing I guess it's already smiling. It can keep transcribing the trip operon and the biosynthetic genes. So that means we will now transcribe biosynthetic genes E, D, C, B, and A. And we will transcribe these when charged trip tRNA levels are low. When those are low, it's because tryptophan levels in the cell are low. So tryptophan levels in the cell are low, charged tRNA, trip tRNA levels will be low, the ribosomes will pause on the leader sequence, loop 2 and 3, the anti-terminator loop will form, RNA polymerase will be able to transcribe the biosynthetic genes, and then trip levels will climb in the cell. Okay, so that's how the attenuation system for the trip operon works. And that is the answer to 6.12.